The presentation, Algebraic Harmony in Genomic DNA Texts and Long-Range Coherence in Biological Systems by Sergei PETOUKHOV. The creators of quantum mechanics, Jordan and Schrödinger, pointed out the key difference between living bodies and inanimate ones. In living bodies, genetic molecules play a dictatorial role. Inanimate objects have no such dictatorial molecules, since they are governed by the average random motion of millions of particles. The study and use of the properties of DNA molecules with their nucleotide sequences which carry genetic information are some of the key tasks of modern science. See the history of quantum biology in this article. Hereditary information is recorded in DNA molecules in the form of a long sequences of 4 molecular letters: adenine A, thymine T, cytosine C, and guanine G. These DNA sequences for each genome of many organisms are presented in the open access and the GenBank in a standard form which gives the impression of a chaotic DNA text of 4 letters with a length in thousands of pages. But as known, these genomic sequences of 4 letters are not at all random. Let's start with an example of the DNA of human chromosome number 1, whose chain contains about 230 million nucleotides, ATCG. Let's first calculate the number of nucleotide A. See below the table. Then in the chain of its doublets, the number of all 4 doublets which start with A, that is AA, AT, AC, AG. Then in the chain of its triplets, the number of all 16 triplets which start with A, AAA, AAT, etc. And so on for each of the set of endplets which start with A. Take here n is equal to 1, 2, 3 till 20. We get the following 20 numbers. It is striking that all these 20 numbers practically coincide with the hyperbolic sequence whose numerator is the number of nucleotides A in this DNA. The deviations of real quantities from hyperbolic sequence are 0.01% and lie in the range 0.03%. Knowing the number of nucleotides A in the genomic DNA text, it is possible to predict with high accuracy the values of 19 other named sums of endplets or oligomers of length n. This new author's method of analyzing and comparing DNA text is called the method of oligomer sums. Similar results are obtained in the same chromosome when calculated the total number of endplets which start with any of the other 3 nucleotides, T, C, or G. The resulting 4 hyperbolic sequences for the human chromosome number 1 differ practically only in the value of their numerators, which are equal to the amounts of corresponding nucleotides. Similar results were obtained for all other human chromosomes, for all chromosomes of all tested eukaryotic genomes, and for all tested prokaryotic genomes. See published numeric results in these articles. If only each K letter for K is equal 2, 3, or 10, or 50 is taken in the DNA text of genomes, then new shortened DNA text with the new sequences of ATCG are formed. it turns out that for each such new text, the same hyperbolic rules for the interconnection of the sums of n-plets are fulfilled with high accuracy. The hyperbolic rules of genomes are fundamentally different from the well-known hyperbolic rule of CIPS, which is not related to information coding and is formulated in linguistics for the percentages of occurrence of individual words in long text. These rules, genomic and CIPS, correspond to different systems of object. Next section, genomes and the harmonic progression. The model how hyperbolic sequences serve as mathematical standards for the general phenomenological rules of oligomer sums in eukaryotic and prokaryotic genomes. These hyperbolic sequences differ from each other only in the magnitude of numerators and their expressions. By appropriate scaling of numerators, each of these genomic hyperbolic sequences is transformed into the historically famous harmonic progression. The name harmonic progression comes from its connection with the series of harmonics in music and with standing waves in a stretched vibrating string. The harmonic progression was studied by Pythagoras, Leibniz, Newton, Euler, Fourier, Dirichlet, Riemann in relation to different mathematical and physical problems. Now the harmonic progression shows itself unexpectedly in all tested genomes. The presented data on the universal hyperbolic rules of the oligomer harmonic organization of eukaryotic and prokaryotic genomes say that these global genomic algebraic invariants remain unchanged over millions of years of biological evolution, during which millions of species of living organisms die and new ones arise, although locally genomic sequences are modified under the action of mutations, the pressing of natural selection, etc. The harmonic progression has interesting mathematical features which can be useful for developing new classes of models of genetically inherited physiological structures. For example, the sums of the first members of the harmonic progression are called harmonic numbers. The series of harmonic numbers serve as the discrete analog of the continuous function of natural logarithm. This in particular connects the harmonic progression with the Weber-Fechner logarithmic law, which is the main psychophysical law and dictates information features for all inherited sensory channels, vision, hearing, smell, etc., whose organs, eyes, ears, nose, etc., very differ each other in appearance. It testifies that genetic and different psychophysical levels of inherited biological informatics are structurally intercorrelated on the algebra harmonic basis. Since the time of Pythagoras, the harmonic progression is associated with harmonics of music and standing waves in resonators. This allows us to believe that the connection between the harmonic progression and the numerical fractal structure of helical DNA texts is of a resonant nature and reflects the existence of a certain system of standing waves. This is consistent with their known hypothesis that helical DNA molecules are helical antennas of electromagnetic waves and therefore, like other helical chiral biomolecules, emit and absorb electromagnetic waves of a certain circular polarization, which provides biomolecules with the ability to exchange radio waves of selective polarization. Helical antennas, including fractal ones, are important in space communications, radars, and cellular telephony. They, unlike other antennas, generate circularly polarized radio waves. They are insensitive to manufacturing defects and violations of the relative orientation of the transmitting and receiving antennas. Chirality of biostructures and circular polarization of their electromagnetic waves are important for pharmacology, physiotherapy, etc. An analog of the helical antenna was discovered recently by Swedish scientists in the tail of the sperm. It is believed that these helices act as their camera to bypass obstacles and search for an egg. Antenna-like helical structures are involved in the biomechanics of coordinated movements. For example, the unicellular organism Mixotrisha paradoxa moves due to 250,000 helical bacteria on its surface, whose helical flagella are coherently being twisted, providing purposeful movement. Electrical and mechanical vibrations in living bodies are closely related, since many of their structures are piezoelectric. Nucleic acids actin, dentin, tendons, bones, etc. Thus, electromagnetic phenomena are accompanied by the phenomena of vibration mechanics. Hyperbolic rules of the oligomer cooperative organization of genomes give pieces of evidence about long-range coherence in biological structures. A possible explanation of this fact can be based on known Froelich theory of long-range coherence in biological systems, which are supported by many authors. In particular, Penrose uses Froelich theory in his consideration of living organisms as quantum mechanical systems. The theory is based on the idea about vibrations in cells, which can resonate with microwave electromagnetic radiation due to a special biological quantum coherence phenomena, which can exist under large energy of metabolic drive. According to Froelich, a phenomenon quite similar to a Bose-Einstein condensation may occur in substances in certain conditions, which are possible for biological bodies. In a certain sense, the mechanism described by Froelich is a special traps of the energy of metabolic processes in a living organism. The son of Herbert Froelich wrote the article Genetic Code as Language regarding quantum coherent states and long-range communication in genomes. Our discovery of the universal harmonic rules of cooperative organization of genomes is an important support of Froelich's theory and popular suppositions about living organisms as quantum information entities. Next section, numerical algebra harmonic mandalas of genomes. Structural analogies of the systems of 4 nucleotide DNA alphabets with the yin-yang schemes of the ancient Chinese Book of Changes and 4-section mandalas have long been noted. Over the course of thousands of years, millions of Buddhists, Hindus, and other believers have created mandalas as a tool of meditation to achieve enlightenment and healing. The creator of analytical psychology, Carl Jung, and his associate physicist Pauli considered the mandala to be an innate archetype of the unconscious and the conjugation of the cosmogram with the psychogram, capable of harmonizing the psyche by bringing all life oppositions to identity. Revealing the connection of long DNA text with the harmonic progression allows us to build for them genomic mandalas of an algebraic harmonic numerical character, in which all sectors and all rings are mutually connected by the harmonic progression. In this numeric DNA mandala, each of the 4 quadrants shows for long DNA the number of N plates which start with A, T, C, or G. The N symbol means on any of the later N, T, C, G. In the normalized mandala of the DNA text on the right, each sector shows the ratios of the numbers of N plates to the number of the corresponding nucleotides. The series of these ratios are universal for all 4 sectors of different genomes and coincides with the harmonic progression associated with harmonics in music. Ancient teachings from the enlightened sages of India and China affirmed the view of musical harmony as something primordial. Buddhists have thousands of years of mandala meditation practice associated with the idea of musical harmony, reflected in the rows of scalable bells in ancient Buddhist monasteries. For long DNA text, in addition to the described mandala of the first order, there are mandalas of the second, third, and higher orders based on plates which start with one of 16 doublets or one of 60 triplets, etc. There is a huge fractal-like hierarchy for DNA mandalas tied to the harmonic progression, considering the conjugation of the later with the harmonics of music. One can think of a whole core of DNA mandalas, which can be turned into ensembles of physical factors for technological purposes. The issues of the affinity of the genetic harmony of organisms with musical harmony are dealt with by our Center for Interdisciplinary Research of Musical Creativity of the Moscow State Conservatory. Conclusions. New materials are received for the development of algebraic and quantum biology. Secondly, Froehlich theory of quantum long-range coherence in biological systems seems to be appropriate for modeling the hyperbolic rules of oligomer cooperative organization of long DNA text in genomes. Thirdly, the discovery of fractal-like phenomena of cooperative organization of long DNA text gives new data for considering helical DNA molecules as electromagnetic antennas with wave of circular polarization. Fourthly, new data about the affinity of the genetic harmony of organisms with musical harmony are received and studied. Thank you very much for your attention.